So for antigen processing, this is a mechanism where we're taking the proteins of the antigen and we're going to break them down into smaller little peptides that can be compatible with either the MHC class 1 or the MHC class 2. And the means of which we go about breaking down that peptide, that's the differences between the two, because obviously they have different structures that they're associated with. The antigen presentation, as its name kind of implies, is just that, presenting the antigen on the host cell surface, be that a skin cell that's infected with, say, I don't know, herpes, or a professional antigen presenting cell such as a dendritic cell. Same thing here. So for MHC class 1 and for class 2, this whole thing kind of starts off in the endoplasmic reticulum and then kind of branches off from there of where they're going to get their antigens from. So in case you remember from general biology, there are these things known as proteasomes. These are compounds or complexes of proteins that play a role in breaking down intracellular antigens, be that a piece of a virus, piece of a mycoplasma, a piece of a protesia that's infected the cell, something that's inside there. But they also play roles in recycling old proteins and breaking down things that don't need to be there. So they have some functions in terms of immunocomplexes, but also functions in terms of just cellular maintenance. Anyway, so they break them down, and they're going to release their peptides into the endoplasmic reticulum, where it's going to bind to the MHC class 1 binding site and then go through the Golgi bodies and as a secretory vesicle end up on the host cell membrane. Over here though, what's going on is we have a endosome coming in. So we've done phagocytosis, right? And we've broken it down, and after we broke it down, the individual pieces, the peptides of it, are going to bind with MHC class 2 and end up on the host cell surface. So they both use the endoplasmic reticulum. It's just that MHC class 2 has to wait a little while longer because he's looking for a specific type of peptides as opposed to that of MHC1. So, in case you didn't know this, viral infections cause an increase in the production of MHC class 1. This is through interferon response. That should be review, I think, for the most part. So, yes, we have proteasome that's always there, that can serve as a immunofunctions as well, but really this is more for maintenance here, as opposed to something known as the immunoproteasome. So once we have interferon gamma being produced, we're going to start making this thing known as an immunoproteasome, which is just like a regular proteasome, but it's better at cleaving peptides into small enough fragments for MHC class 1. Because remember, for MHC class 1, the peptides can be 9 amino acids. That's the longest. That is the maximum length we can have. So we have to have something that's really specific and is going to cleave things that are going to fit just right. In case you didn't remember this, on the outside of a proteasome, there are these two things here known as the 19S cap, but I like to think of it as kind of like the Sarlacc pit from Star Wars, you know, the little like tentacle things. I don't know if you ever played Battlefront, but anyways, on the outside here, these things are involved in shuffling the proteins. They're going to grab the proteins and they're going to shuffle them to the interior. Then once they're inside, this right here, all these beta subunits that you're seeing here, or the LMP and the MECs, these are both just catalytic subunits, or I guess you could call them proteolytic if you wanted to be really specific about this. And the same thing for this outside here, these are the tendrils, I like to think of it. They're designed to bring stuff into, that are things that are flying out here. They're going to grab them and throw them in here, and then once they're going through there, they're going to be grinded up and broken down into the individual ones here. The only difference is, if we could just clear all this, the immunoproteasome is, again, like I said, more effective. Okay, so processing of cytosolic proteins into the peptides for transport into the ER for MHC1. So we've broken down the peptides. Now how do we get them into the endoplasmic reticulum so that they can be associated with MHC class 1? The way that we do this is through the use of an enzyme known as TAP, and this is a active transport enzyme. So we're hydrolyzing ATP, we're using ATP. This must be an important process if we're going to waste precious ATP. It's a heterodimer, which is why I kind of hate this picture, because it shows them kind of almost looking, it looks like a homodimer from here, but it's actually a heterodimer of TAP1 and TAP2. That's not superbly important so much as you understand that it's using ATP to pump peptides into the endoplasmic reticulum. And so let's talk about what's going on inside the endoplasmic reticulum before this point here. So the first thing that's happening is we have MHC class 1. Remember we said that these are all the alpha. These are all expressed as a part of the same gene. They're probably all translated as together. But beta 2 microglobulin has to bind, non-covalently at least, for MHC class 1 to be stabilized, and it also has to bind for MHC class 1 to undergo a conformational change, which is going to make that active site more susceptible to antigen binding. But to stabilize this conformation here, until this guy binds here, calnexin comes in, and it's going to go ahead and just bind to a similar site. You could also think of calnexin has other roles, but it is a chaperonin, holding that protein in the appropriate three-dimensional place. So once beta 2 microglobulin comes in and binds, conformational change is going to occur. The active site is going to become a little bit more receptive. Calnexin is going to leave. So calnexin is going to be released, and the heterodimer of class 1 heavy chain and beta 2 microglobulin are going to form the peptide loading complex with the helps of calreticulin, tapicin, TAP, ERP, and then PDI. It's interesting that my textbook doesn't talk about PDI, but it must not be that important. If there is a function for it, I don't know it. Sorry. Tapicin, however, has a very important functions. Calreticulin have very important functions as well, and then ERP. So I guess if I were to try to walk us through here of the processes that are going up here, here's the tapicin. Tapicin is going to serve as a bridge molecule, if you could think of it as that way, and it's going to ensure that the MHC receptor site is going to receive the proteins from the TAP. So it's making sure that they get into the actual binding site there. For the ERP57 or ERP57, it's just a very weird name. Some interesting fun facts about that. This is a thiol reductase, and what this does is it's going to stabilize things by binding, and it's going to protect the disulfide bonds of everything from being degraded. To me, this reminds me of preparedin, because they will have that similar curve-like structure, the way that they draw them. I know this is a horrible representation of it, but I don't know why. That's just what it reminds me of. Now, for calreticulin is actually just a soluble chaperonin. So it's doing the same thing of keeping this thing stabilized until it binds to the actual peptide that it's going to be associated with, it's relatively speaking kind of unstable. So we have to have helpers to come in here. So we now have this complex here that's creating an extremely favorable conditions. It's very closed spaces here. That's not a lot of room for those peptides to go. So of course the peptides are going to come in and bind. So peptide is delivered by TAP, binds to the class 1 heavy chain, forming the mature MHC class 1 molecule. And once this binds, it's going to undergo a conformational change that makes it way more stable and doesn't need the help of calreticulin or all the other sub-complexes that are associated with it. And everything's going to kind of dissociate off, and it's going to leave the endoplasmic reticulum, go to the Golgi bodies, and ultimately end up on the surface membrane of your host cell. Okay, so we didn't really talk about this other thing here though, but there's this stuff called ERAP. The name for ERAP, if I were to, I'd written it down and I wanted to read it because it's I swear to God, these the acronyms are just stupid as hell to me. Endoplasmic reticulum aminopeptidase. That's actually kind of a helpful one. But anyways, so say you have a peptide that binds here, and that peptide is great. It's super compatible. It's really stable. But the problem is that it's too big to fit into the active site and too big to leave the nucleus. So ERAP will come in, and it will cleave it, as its name implies, one amino acid at a time until it's down to a small size enough to where it can be excreted. And this is very helpful because remember, MHC class 1 molecules have very small peptide loading sites. Okay, so the only point that this slide is trying to show is that it happens in different intracellular compartments. Okay, so processing occurred in the case of MHC class 1 in the endoplasmic reticulum is when we got all this stuff. In the case of MHC class 2 though, it's happening in the endosome. And that's what we're dealing with here. But at the end of the day, they both start off at the same locations. They both have similar functions. So for MHC class 2 molecules in the ER, the peptide loading is taking place in known as cytosolic vesicles, if you could think about that, and presentation for the helper T cells, the CD4 positive T cells. So the antigen is going to be taken in through the intracellular up in an intracellular vesicle, undergoing phagocytosis, fusion with a lysosome, pHs, things like that. Proteases are going to come in, they're going to cleave it, they're going to break it down. This is all reviewed to you, hopefully. And then vesicles containing the peptide are going to fuse with the MHC class 1 molecule. So this is what's going on at the big picture thing here. This is stuff you should be familiarized with. However, the process of how this works is a little bit kind of similar to what we talked about there. So the invariant chain is something that's kind of involved in this, and it's not that the MHC class 2 molecule is necessarily unstable, so much as it is that we want to make sure that it has the right peptide binding at the right point in time. So as it's going through the endoplasmic reticulum, we don't want it binding to those peptides because that wouldn't be helpful for it. So the invariant chain is going to go ahead and come in here. It's going to block that access there by all other peptides, and it's also going to help stabilize it to a lesser extent. Now, in the vesicles of the invariant chain, once they leave the endoplasmic reticulum, they're going to start being broken down into something known as a clip fragment. This clip fragment is basically just holding in the active site, so that, again, it's keeping it stable, but also to make sure that nothing else comes in there and binds with it. So clip is going to block the binding of peptides from MHC class 2s in vesicles. And then, so we want to make sure that we have this peptide binding at the exact appropriate time. Something known as HLA-DM in humans is going to facilitate the release of clip, which once clip is gone, well, the active site is now available to bind to peptides, right? And so it's going to then, binding takes place at the last second, if you can think of it, and then it goes off to the endoplasm, or to the host cell membrane. Okay, so T cell receptors are specifically designed to recognize both the peptide and the MHC molecule. I've stressed that, I think, already. But there's the, so here we see the T cell receptors interacting with the antigen sites of the peptide as well. Also, though, it's the T cell receptors themselves, not just the co-receptors, but also the T cell receptors themselves. And I think that's really interesting. This is a really cool diagram showing a unique three-dimensional structure. Here's the peptide of an MHC class 1, literally in the satchel part. And these are the hypervariable loops of the T cell receptor, and the alpha and then the beta. So crazy to me. But a lot of work probably went into that. So here's something else that's interesting. Say, for example, that I wanted to mount an immune response against a virus, and I didn't want to sit around and wait until it infected all the cells in my body. I wanted my professional antigen-presenting cells to do this thing so that we could actually get antibodies against this. So we have to have ways of expressing intracellular pathogens on dendritic cells or macrophages, right? Because I don't know about you, but I would not just like the idea of CD8 T cells doing everything by themselves. We need antibodies to come in and work as well. So not all antigen-presenting cells are going to be infected by a particular virus, right? Hepatitis C virus is not going to infect your dendritic cells. So the phenomenon of actually how this works is pretty, it's observable, right? We can make antibodies to intracellular pathogens, right? Yeah, viruses. This is we've been knowing this since the days of Edward Jenner. But the actual mechanism of this, we really don't understand in too much detail. There are, however, two hypotheses that are currently accepted for, or not accepted, but are being debated right now for how this process works. The first one is that viral components are going to be delivered from the endocytic vesicles into the cytosol for degradation by the proteasome and then presentation by the MHC class 1. This I can't speak on behalf of the evidence to support these claims, but the other one, though, is that phagocytosis of a cell is going to allow complexes of MHC class 1 molecules and viral peptides to be transferred from, say, for example, the dead hepatocyte or the dead skin cell or whatever it was that they ate, and there's going to be transferred, they're going to stay in that conformation and then end up on the MHC class 1. And that's what this picture is showing us. This is the macrophage, for example, eating the dead virally infected cell, and then that's going to end up on the, this is a MHC class 1 protein from that virally infected cell that ended up being presented on here. This, to me, would be very easily verifiable with a GFP that you could attach to the MHC class 1, confirm that it is being expressed on here, and then if you saw it in here, well, it'd be a safe bet to make that it was compatible with that. But anyways, that's